Section 1. The Building Blocks of Life. An Introduction to Proteins. Proteins are the workhorses of life. They are fundamental to the structure, function, and regulation of every cell in our bodies. From the keratin in our hair and nails to the hormones that orchestrate our growth, proteins are involved in virtually every biological process. They are large, complex molecules, composed of chains of smaller units called amino acids. These amino acid chains fold into intricate three-dimensional structures, which determine the protein's specific function. Proteins are essential for a vast array of biological processes. They act as catalysts, speeding up chemical reactions within cells. They transport molecules like oxygen in the blood. They provide structural support, giving cells their shape and enabling movement. They also play crucial roles in immune defense, cell signaling, and DNA replication. The diversity of protein function is truly remarkable, reflecting the complexity of life itself. Understanding protein structure and function is crucial for unraveling the mysteries of life. By delving into the intricacies of protein science, we gain insights into the molecular mechanisms that govern our bodies. This knowledge paves the way for developing new therapies, diagnosing diseases, and engineering novel proteins with specific functions. As we unlock the secrets of proteins, we unlock the secrets of life itself. Section 2. Amino acids, the foundation of proteins. Amino acids are the building blocks of proteins. There are 20 different amino acids, each with a unique chemical structure. All amino acids share a common backbone, consisting of a central carbon atom bonded to an amino group, a carboxyl group, and a hydrogen atom. What distinguishes each amino acid is its side chain, also known as the R group. The side chain varies in size, shape, charge, and chemical reactivity, giving each amino acid its distinct properties. Amino acids can be classified into two main categories: essential and non-essential. Essential amino acids cannot be synthesized by the human body and must be obtained through our diet. There are 9 essential amino acids: histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Non-essential amino acids, on the other hand, can be produced by our bodies. These include alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, and tyrosine. The classification of amino acids based on their side chains is crucial for understanding protein structure and function. Some side chains are hydrophobic, repelling water, while others are hydrophilic, attracting water. Some are acidic, carrying a negative charge, while others are basic, carrying a positive charge. These chemical properties influence how amino acids interact with each other and with the surrounding environment, ultimately determining the protein's three-dimensional shape and function. Section 3. Peptides, small chains with big roles. Peptides are short chains of amino acids, typically containing between 2 and 50 amino acids. They are formed when amino acids link together through peptide bonds. Peptide bonds are covalent bonds that form between the carboxyl group of one amino acid and the amino group of another. Peptides play diverse roles in the body, acting as hormones, neurotransmitters, and signaling molecules. One well-known peptide is insulin, a hormone produced by the pancreas. Insulin regulates blood sugar levels by promoting the uptake of glucose by cells. Another important peptide is oxytocin, often referred to as the love hormone. Oxytocin is involved in social bonding, trust, and maternal behaviors. It is released during childbirth and breastfeeding, strengthening the bond between mother and child. Peptides also play a crucial role in the immune system. Antimicrobial peptides, for instance, are small peptides that can kill bacteria and fungi. They are part of the body's innate immune defense, providing a first line of protection against infection. Other peptides are involved in regulating inflammation, wound healing, and blood pressure. The diversity of peptide function highlights their importance in maintaining overall health and well-being. Section 4. The architecture of proteins, from primary to quaternary structures. Proteins are not simply linear chains of amino acids. They fold into intricate three-dimensional structures, which are crucial for their function. The specific shape of a protein is determined by its amino acid sequence and the interactions between the amino acids. There are 4 levels of protein structure: primary, secondary, tertiary, and quaternary. The primary structure of a protein is simply the linear sequence of amino acids. This sequence is determined by the genetic code, which dictates the order in which amino acids are linked together during protein synthesis. The secondary structure refers to local folding patterns within the polypeptide chain. These patterns, such as alpha helices and beta sheets, are stabilized by hydrogen bonds between the backbone atoms of the amino acids. The tertiary structure is the overall three-dimensional shape of a single polypeptide chain. It is determined by interactions between the side chains of the amino acids, including hydrophobic interactions, hydrogen bonds, ionic bonds, and disulfide bridges. The quaternary structure refers to the arrangement of multiple polypeptide chains, or subunits, that come together to form a functional protein. This level of structure is stabilized by the same types of interactions that stabilize tertiary structure. Section 5. When proteins unravel. Denaturation, cofactors, and the world of enzymes. Protein structure is not static. It can be altered by changes in temperature, pH, or exposure to certain chemicals. When a protein loses its three-dimensional structure, it is said to be denatured. Denaturation can disrupt the protein's function, rendering it inactive. For example, cooking an egg denatures the proteins in the egg white, causing it to solidify. Some proteins require non-protein components called cofactors to function properly. Cofactors can be inorganic ions such as iron or zinc, or organic molecules known as coenzymes. Coenzymes are often derived from vitamins. For example, vitamin B12 is a precursor to the coenzyme cobalamin, which is essential for certain metabolic reactions. Cofactors bind to proteins often at the active site and participate in the protein's function. Enzymes are a special class of proteins that act as catalysts, speeding up chemical reactions without being consumed in the process. They are essential for virtually every biological process, from digestion to DNA replication. Enzymes work by lowering the activation energy of a reaction, making it easier for the reaction to occur. They do this by binding to the reactants, or substrates, and bringing them together in a way that facilitates the chemical reaction. Section 6. Enzymes in action therapy, diagnostics, and the power of inhibition. Enzymes play a vital role in medicine and diagnostics. They are used in a wide range of applications, from diagnosing diseases to developing new therapies. For example, the enzyme lactate dehydrogenase is used to diagnose heart attacks, as elevated levels of this enzyme in the blood can indicate tissue damage. Enzymes are also used in drug development, as they can be targeted by drugs to inhibit or enhance their activity. Enzyme inhibitors are molecules that bind to enzymes and reduce their activity. They can be competitive inhibitors, which bind to the active site of the enzyme, preventing the substrate from binding, or non-competitive inhibitors, which bind to a different site on the enzyme, altering its shape and reducing its activity. Enzyme inhibitors are used in a variety of medications, including Viagra, which inhibits the enzyme phosphodiesterase 5, leading to increased blood flow and facilitating erections. Enzyme regulation is essential for maintaining cellular homeostasis. Enzyme activity can be regulated in a number of ways, including allosteric regulation, covalent modification, and feedback inhibition. Allosteric regulation involves the binding of a molecule to a site on the enzyme other than the active site, which can either enhance or inhibit the enzyme's activity. Covalent modification involves the attachment or removal of a chemical group, such as a phosphate group, which can alter the enzyme's activity. Feedback inhibition occurs when the product of a metabolic pathway inhibits an enzyme earlier in the pathway, preventing the overproduction of the product. Section 7. The Symphony of Enzyme Activity, Regulation, Kinetics, and the Future of Medicine. Enzyme kinetics is the study of the rates of enzyme-catalyzed reactions. The rate of an enzyme-catalyzed reaction depends on a number of factors, including substrate concentration, enzyme concentration, temperature, pH, and the presence of inhibitors or activators. Understanding enzyme kinetics is crucial for optimizing enzyme function in various applications, from industrial processes to medical treatments. A key concept in enzyme kinetics is the Michaelis-Menten model, which describes the relationship between substrate concentration and reaction rate. This model allows us to determine the enzyme's kinetic parameters, such as its Michaelis constant, Km, and its maximum velocity, Vmax. Km is a measure of the enzyme's affinity for its substrate, while Vmax is the maximum rate of the reaction when the enzyme is saturated with substrate. The study of enzyme kinetics is fundamental to our understanding of biological processes, as it provides insights into how enzymes function and are regulated. This knowledge is crucial for developing new drugs, optimizing industrial processes, and advancing our understanding of human health and disease. As we continue to unlock the secrets of enzyme activity, we pave the way for new discoveries and innovations that will transform medicine and improve human lives.